start and let's share the screen. Where's the screen? Yep. Okay. So let me know if you are able to see the screen, just to make sure. Thumbs up. Or okay. Xuan, you can see. Eric, can you see? At least you can see. Eric, you can see? Yep, good. So the starting time is 8:35 a.m. Right. All of you have attended my class, so you know what to expect. What is the process? The process is the 10-step process, and we'll start with step number 1. Right. Before that, let me mention a few things. Number 1 is this is not a normal tuition class that you'll be expecting that I'll be speaking 80% of the time. In fact, the work that is being done is mainly on your side. You have to read. What my role here is to facilitate you to follow through the step 1 until step 10. And in fact, most importantly is my main objective is not to teach you biology or physics or chemistry, but my main objective is to teach you this new study method that works for me. And I hope by learning for you, by teaching you this learning method, you'll be able to learn and study more efficiently, right? So as you know, within this 2 to 2.5 hours, you'll be able to have a good understanding on this chapter. And I believe this is a good result because in 2 to 2.5 hours only, you can have a good understanding of one chapter. And obviously you have more time to do other things, whether to continue with your YouTube or whether to continue with your TikTok or continue to reinvest into revisions or another chapter, right? It's your choice. It's up to you already because you have more time than you had before. Once you are able to master this way of learning and if it works for you, right? So let's start now. Today, the chapter is biology. It's about cells. And step 1, as usual, we are going to create drawers. For this particular chapter, the name is cell. We are going to create 5 drawers. You can see in front of you, the screen, there are observing cells, plants and animal cells, specialized cells, movement of substances, unicellular organisms. To remember this, I created this very short story. Let's take, okay. I cell, plant and animal. Okay, I sell 2 things, plant and animal. And that animal that I'm selling, is specialized in moving one organ, right? Sounds a bit scary, right? That animal, whichever it is specialized in moving one organ, whichever organ. It could be your liver, it could be your lung, it could be your eyes, it could be your heart. So what does it mean? I sell, the cell sounds like observing cells, right? So I sell to represent drawer number 1. Plants and animals, which is plant and animal cells. And specialized is specialized cells. Movement of substance. One organ, unicellular organism. Uni is one, right? Organism, so I link it with organ, right? Make it a bit scary. The animal is a bit, a bit creepy and animal, where the animal job is to move one of your organs, and that's his specialty, right? So if you can get a pen or paper, if you have, if you don't, it's fine. And you write down these few words, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, only 11 words in front of you, because you need this, because I'm going to ask you questions, which tell me drawer number 1, number 2, number 3, number 4, number 5. Right, then you can just refer to this easily, and it will help you to remember. Right, I think by now you know this study method is all about anchoring your brain into something that is easy for you to remember. That's why I created the drawers. So the drawer is the anchoring point. All right. So I hope you get your pen and paper and write this down. It's only 11 words. Now let's continue on step number 2. Okay, anyway, I'll give you 30 seconds. 30 seconds to write it down. If you want. If you don't, then just wait for 30 seconds before we proceed to step 2. Oh, I'm checking my message. Okay, no important message. Okay, 5 more seconds. Right. Now, step 2, read keynotes. So these are the keynotes. It's not a lot. Okay, let's take 2 minutes to read. I think 2 minutes is more enough. And don't be worried and anxious if you do not fully understand what you're reading. That is fine, and that is normal. Yeah, do not need to read the words inside the brackets. These are the stories. Later we will use it later as in the later stage. Okay, 25 seconds. All right. So what you have read just now is written in 5 drawers. Right. You have the drawer number 1, observing cells, plant and animal cells. Drawer number 2. Number 3 is specialized cells. Number 3 is movement of substance. Number 4 is movement of substance. Number 5 is unicellular organism. Right. So you need to know in drawer number 1, there's something about microscope that you'll be learning. And this famous scientist 350 years ago. Then in drawer number 2, you'll be learning components on plant and animal cells and the difference between plant and animals. Specialized cells, what are the specialized cells that is important for animal and plants that you need to know? Then how the movement of substances, basically it's the diffusions in the process of respirations, gas exchange, water absorptions, etc. Then there are 2 unicellular cells. Human, like us, we are having billions or trillions of cells. And in this world, there's organism that is having 1 cell only. Right? We're going to learn about that later. Right? So there are 2. amoeba and euglena. Yes. So let's go to step 3. Right? Step 3, we'll have this part. Okay, let me see how long is this. Okay, so we divide into 2 parts, 1 minute and 1 minute. You can read from observing until seen, 1 minute. And how do you read this? This is drawer number 1. So in drawer number 1, you see this is observing cells, and you have Robert Hooke, and you have microscope. So this, oh sorry, and number 3 is total magnification. Let's bold it. So when you were reading this just now, maybe you do not have full understanding what is this. But in step 3, it gives more elaborations. So hopefully that with this more elaboration and the picture here, you'll be having deeper understanding on what was in this drawer, in this step 2, in this one small line over here, right? So let's start for 1 minute to read from this drawer 1 until it can be seen. Start now. All right. Let's continue here. Okay, Jaden is in. Let's continue from microscope usage magnifications until the 400. 1 minute. Jovin and Jaden, can you see the screen? We are sharing the screen.
Yes.
Okay. Okay, good. Right. So this is drawer number 1, observing cell. Observing cell in the story is I cell. I cell. Okay. And what is the main point or main message in this drawer number 1 is to learn about microscope. Right. Learn about microscope. So you see in step 2, we have some key words related to the component of microscope. And also, how do we calculate the total magnifications of the microscope? And in step 3, on your left side, there's a little bit more elaborations on these microscope components over here. And also the usage and magnifications of the lens on microscope. If you are able to fully understand, if you think that you are able to fully understand microscope by now, by just reading step 2 and step 3, excellent. If not, it's fine, because we are just 10 minutes into our class, or 10 or 15 minutes into our class. So don't worry that you do not fully understand everything that you read. You're going to understand it as we progress along, right? Okay, now we are in drawer 2. Now we're in drawer 2. So let me see how far is this. Okay, so we divide into 2 parts. From drawer 2 until the sun, 1 minute. Ten seconds. All right, let me try to guess what is your feeling now after reading this? You'll be thinking, how can I remember this? Nucleus, cell membrane, cytoplasm, mitochondria, cell wall, buccal, and chloroplast. I've got you a solution, right? And this is the story. This is the main story of drawer number 2. New mom, imagine now you have a new mom, not the mom that you have. And what is she doing? Cooks meal for you with green vegetable, something that you do not like. I think most of the students here, they don't like vegetables. Yeah, if you do, then congratulations. But if you don't, yes, this what I'm trying to trigger the emotions, right? You have a new mommy and she's cooking vegetable and curry, spicy curry for you, right? So new mom, new is with the N, nucleus, cell membrane. I did not use cell, I use membrane for mom. Cook cytoplasma, meal mitochondria, with, I did not use cell, I use wall, W, and then buccal is veggie, curry is chloroplast, right? So this is to help you to remember easily or easier, right? So you have this nucleus, membrane, cytoplasma, mitochondria, wall, buccal, and chloroplast, right? Then what you need to focus now is what is the function of these components, okay? And let's go to the next one. From the membrane here until structure, 1 minute. Ten more seconds. Done. So this is drawer number 2. The main idea of drawer number 2 is to learn about the plant and animal cells, the component in it, and which component plant cell is having more than animal cell. So these 3 components, the last 3 components, right? The with veggie curry. Let's move to drawer number 3. Drawer number 3, let's see how far is this. Yep, a little bit more. Okay, let's do this part. specialized animal cell in 1 minute. Sorry. Yep, like this. Right, so let's move to the second part. In drawer 3, specialized cells for plants. Okay, also I'll give you 1 minute just in case you need more time to continue the animal part, which is the upper part. Ten more seconds. Okay, these chapters tend to, we need to memorize a few new terms. So I created stories at the early stage in step 2, so you can remember it easier. Right, so they are animal and plant specialized cells, which are the nerve cell, red blood cells, sperm cells, egg cells. Then for the plant, you have the leaf and also the root hair. So how do I remember this easier? Is I created a story. Just imagine a sperm, and that sperm is bloody. It's looking for eggs, so it's searching for eggs, but accidentally left his hair. Right. So it's not a logical story, but it's silly enough for you to remember this easily. So imagine this sperm is looking for eggs, and this sperm is bloody, is having a lot of blood on his body, but accidentally left his hair at home. All right. So bloody represent the red blood, sperm represent the sperm cell, needs represent the nerve, eggs represent the egg cell, left represent the leaf, and his hair represent the root hair cell. Okay. So let's move to drawer number 4. Drawer number 4 is movement of substance. It's basically about diffusions. Let's start now. 1 minute. Okay, 5 more seconds. Done. I think this explained by itself. If you able to read and understand this, I think it's self-explanatory, right? If not, don't worry, we are still in step 3. Step 4, step 5 will make you understand better if you do not fully understand it. All right, we are in the last drawer. Last drawer, let's divide into 2 parts, I think. Let's 2 parts. Okay, let's do this first part first. For 1 minute. Okay, 10 more seconds. About amoeba. Okay, let's learn the second one. Only one cell. They only have one cell. The second one is euglena. Let's start now. Twenty seconds. All right. The story for this drawer number 5 is Eugene and Angmo. Eugene, if you have a friend called Eugene, yes, so just imagine this friend is having one cell. Angmo, Eugene is obviously sounds like euglena, and Angmo sounds like amoeba. Angmo for Xuan, I think she knows what is this. I'm not sure whether Jaden should know what is this. Elise, I think Joanne should know definitely. Eric, I'm not sure whether you know what is this Angmo. Do you know what is Angmo? Angmo is like a Hokkien word to describe Westerner, right? If not, then just remember Angmo as another friend, right? I think this will be yeah, this is representing amoeba. All right. So before we go into step 4, let's read this again. Step number 2, and tell me if you tell me, do you feel that by reading this again, now you can understand step number 2, the content in step number 2 better? Right. Let's take 1 minute to read. 1 minute to read. Here, step 2. 20 seconds. Later, I'm going to ask you the questions. I think most of you know what is the question I'm going to ask. Okay, can someone tell me what is the story that we created just now to help you to remember the drawer? Okay, Eric, let's start with you.
I sell plants and animal that specialize in moving one organ.
All right. So, anyone else can tell me what is the name of the 5 drawers? Elise, Jovin? Eric again? All right, let's do Eric.
Observing cells, plant and animal cells. Oh wait, observing cells, plant and animal cells, specialized cells, movement of substances, and unicellular organisms.
Right. All right. So this chapter is about these 5 drawers. And how do you remember these 5 drawers is exactly what Eric mentioned just now, the story. Just imagine now you're selling 2 things. One is the plant and one is the animal. And that animal, he's special in doing what? Moving one organ. That specialized in moving one organ, right? Moving somebody's eyes, moving somebody's liver, moving somebody's nose, right? And that will help you to remember these 5 drawers. And I believe and I hope that by now you read this step number 2, you can understand the content of step number 2 better. In 2 ways, look at it into 2 angles. One is there are 5 drawers. And now you understand that there are, for drawer number 1, then you break down into this 5 drawer. Drawer number 1, you need to understand a bit more about the microscope and the magnification. Then drawer number 2, you have this new mom's cooked meals with veggie curry, right? And is to be able to explain the cell components for the plants and animals. Then the specialized cells, which is the drawer number 3, also there is a story in it to help you to remember the specialized cells that is in plant and animals. Bloody sperms needs eggs, accidentally left his hair at home, right? Drawer number 4, movement of substance. So this is the diffusion process. We'll go into the story later, because if I tell you now, maybe it doesn't make sense. So unicellular organism. So there are 2 guys. These 2 guys is amoeba and euglena, Eugene and Amo, right? And you need to know a bit more about them. What is their shape? How do they move? How do they eat? And the structure, the key structure in their bodies, right? So that is the framework, right? So now next step is step number 4, right? Step number 4. Let's do this. We'll take 1 minute to read this. Step number 4. How do you do this? Read the question. Ask which drawer is this. Then read the answer and use this as a reference. Use this as a reference. Because like I say, our brain needs an anchoring point. So the drawer is one anchoring point, and the keywords inside this reference are also the anchoring point. Right? So read the question. Ask yourself, what drawer is this? Drawer 1, 2, 3, or 4, or 5. Then read the answer and use this as anchoring point. Let's start. 1 minute. Now, this is obviously drawer number 1. Read until total magnification. Here. Oh. 1 minute is up. Right. Let me come back. Okay, where's this? Close this. Yep. Let me come back to this drawer and explain about these stories. Robert eyeing the lens on the stage. So there's a stage in front of the robot, and there is 2 lenses there. And Robert focused twice in eyeing the lens, and then run at the speed of light towards the lens. Right. So Robert eyeing the lens on stage, focus twice, run at the speed of light. This is to help us to remember the key components in microscope. Eyepiece, which is eyeing. Objective lens, which is the lens. Stage, obviously is a stage. Then coarse focus and fine focus. So focus twice. And light. Right. So Robert wanted the lens, and the lens is right on top of the stage. So he is eyeing the lens, and he needs to, before he runs at the speed of light, he needs to focus twice to make sure that he's running towards the lens and get the lens that he wants on the stage. Right. So Robert eyeing the lens on stage, and he focused twice and run at speed of light. So eyeing is the eyepiece, objective lens, stage. Then there are 2 focus for the microscope, coarse and fine, and light, right? So let's go to question 2. Question 2, do the same thing. Ask yourself, which drawer is this? After reading the questions. 1 minute. Let's start now. Ten seconds. Done. If you need more time, let me know. Right. If not, then we will continue at this pace. So this is drawer number 1, total magnifications. It's talking about the eyes piece lens magnifications and objective lens magnifications. How do you calculate them? It's very easy. It's just a simple multiplication equation. FAQ 3, question number 3. By the way, we have 8 questions. After the 8 questions, then we have a break. Toilet break and water break. So we have 7 more questions to go. So let's start now. Oops. This is drawer 2. your new mum with the new curry. All right. Now we're moving to question 4. Question 4. Question 4. Let's divide into, let's do 1.5 minutes. Right. This is draw number 2. And it is about the new mum also. New mum with your favorite vegetable curry. Start now. Okay, 40 more seconds. Five more seconds. All right. Question 5. Okay, let me see. How should we do this? Okay, we will do 1.5 minutes also. And I'll scroll down later. This is drawer 3, specialized cell. One more minute. Thirty more seconds. Remember, this is the bloody sperm needs eggs. Accidentally left his hair at home. You know, this boy called sperm, and this eggs called this girl called eggs. The sperm is chasing after the girl. Right? Okay, question 6. Let's do also 1.5 minutes. This is drawer 3, specialized cell for plants. 30 seconds. Five seconds. All right. This is the hair that the sperm left at home. Question 7. This is movement. Movement is drawer 4, diffusion. Same 1.5 minutes. Let's do it now. Thirty seconds. All right. Now I hope you understand drawer 4 better than 2 minutes ago. Okay, this is the last one before we go for a water break. So let's do 1.5 minutes. This is obviously the drawer number 5. The one cell guy. One more minute. Thirty seconds. The two one cell guy. The Angmo and the Eugene. 10 seconds. All right. As promised, we're gonna have 5 minutes water break or toilet break. Let's see you after 5 minutes. 30 seconds. Okay, can I have a show of hand? Who is back? Eric, Strand. What about Elise? Jovin is back. Elise and Jordan. Are you back?
Yes.
All right, everyone is back. Okay, I hope you are following so far. We started at, what time did we start? We started at 8:35. Now it's 9:29. It's about 1 hour. I hope you are following so far in this 1 hour. And let's do a quick review. A quick review on the 5 drawers. You have this story. You are selling 2 things, 2 items. On your left is the plant. Imagine that on your left is a plant, and on your right is an animal. And that animal is specialized in moving one organ, right? So there's these 2 items. And how does this help you to remember the drawers? Observing cell, because I sell. And you sell what? Plant and animal cells. And that animal is specialized cells. And movement of substances. Unicellular organism, moving one organ, right? So by now, if you read this again, step 2, just glance it through quickly. I believe and I hope that you understand it way better than 1 hour ago. Right, you know that drawer number 1, you need to learn, or you have learned something about the microscope components. How do you use the microscope, and what is the magnifications formula they are using in the microscope? By remembering this very short story, Robert eyeing the lens on the stage, right? He wants to get this lens, and this lens is on the stage. He's running towards the lens, the stage, and before he runs, he focused twice to make sure that he's at the right direction, and then he ran at the speed of light, right? So this story can help you easily to remember these components. You don't need to remember this and write it down for 200 times, right? You can remember it quite easily now, right? Robert eyeing, which is the eyepiece, and the lens is an objective lens on the stage, which is stage. And you focus twice. The robot focus twice. So it's a coarse focus and the fine focus, and it's run at the speed of light, right? So why Robert? Because Robert Hooke. All right. Then drawer number 2 is also relatively easy. You have a new mom now, and she cooks your meals with your favorite vegetable curry, right? Then specialized cell. Oh, okay, there's a boy and there's a girl, and the boy is chasing after this girl. She likes her, and accidentally he left his hair at home. So bloody, and the boy is covered in blood for whatever reason, I don't know. All right. So bloody sperm needs egg, accidentally left his hair at home. Right. So that's to represent the bloody. The bloody is to represent the red blood cell. Sperm cell, needs nerve cell, egg cell, leaf cell in plant, hair, root hair cell in plants. Movement of substance, the diffusion. Right, movement of particle from high concentration to low concentration. Right. Let's take one very funny story. Right. I think everyone farted before. Right. When you farted, and then somebody next to you will smell that the bad smell. This is also part of the diffusion. So it was high concentration, and then it was diffused to a low concentration to somebody's nose. Right? Okay. I hope this example can help you to remember diffusions better and easier. Right? So diffusion is not only valid for this example, this FAT example, right? It can be available. It is also using in other processes like respirations, plant gas exchange, water absorption, which you read in step 4 just now. In step 3, if you still haven't get hold of the full understanding about this, don't worry. You still have step 5, and you still have step 6, and 7, and 8, and 9, right? Okay, then the last drawer, Eugene and Angmo. Okay, this explained by itself, right? Amoeba and Euglena. All right, so let's do another heavy reading, which is read the guides, right? So we'll do the same drills. You read, before you read, you guess which drawer is this, but this one is obviously drawer number 1. And then try to intentionally identify if there's any new information here that is not here or you have not read before in this 1-hour class that we attended. Right, let's see how we want to break this down. We'll break it down into 2 parts, I think. Okay, we'll break it down into drawer 1 until precise focusing. Right, this is the microscope. And if you translate it into story, this is the robot eyeing the lens on the stage and focus twice and then run at the speed of light. Okay, let's do 1.5 minutes for this part. Drawer number 1. Let's start now. Ten more seconds. If you need more time, let me know, please. All right. This is drawer number 1 related to the microscope. All right. So the usage of the microscope and the magnification process. So that is the second part. Let's do 1.5 minutes. 10 more seconds. All right. We have done drawer number 1. Let's move on to drawer number 2. Let's see how much content it is. Okay. Let's start with 1 minute here. Remember the story? Your new mom is cooking something for you, your favorite vegetable curry. All right, let's start now. All right. Second part of drawer number 2, plant and animal cells. Plant and animal cells. Right, this part. Let's do 1.5 minutes. 90 seconds. Thirty seconds. All right. I hope you like your green vegetable curry. All right. Next one. Okay. This one small part. All right. Let's do this in 1 minute. This is the sperm and also the egg story. And he left his hair at home. Okay. Second part of this drawing number 2, which is the part that the hair and the hair of the sperm. Let's do 1 minute. Okay. Next drawer. Drawer number 4. The movement of substances. Movement of substances. Okay. So we divide into 2 parts. Let's do this one. 1 minute. All right. Diffusion can happen in liquid or gas, not solid. Liquid or gas. Okay, this one also 1 minute if you need to read again, or if you did not finish reading just now. All right. So I think this story maybe makes sense now if I tell you now. Glucose is a boy or a girl. He breathed to exchange gas, like everyone does. We breathe and we exchange gas, oxygen to carbon dioxide. And then he drinks water and sleep. Right. So this is to anchor the key message of glucose for respirations, and then gas exchange, and then water absorption, and sleep. Sleep is to represent wilting. So imagine your friend called glucose, he's breathing. And why he's breathing? Because he wants to exchange gas. And at the same time, he's drinking water. Then he went to sleep. All right, the last drawer. Last drawer. Okay, let's divide it into 2 parts. This part, 1 minute. Okay, let's go to this part. Let's do 90 seconds. 30 seconds left. Ten seconds. All right. Now, create stories. We have already done this indirectly, but now let's do this. Let's do this now again. So there are 5 drawers: observing cell, plant and animal cells, specialized cells, movement and substance, movement of substances, unicellular organism. So the story, we have talked about this like a few times just now. Robert is eyeing the lens, you see. If you can understand my drawing, by the way, I'm not an artist, as you can see. So this guy is Robert, and is eyeing the lens at the stage, on the stage, and he focused twice. Focus it, first time, second time. Then he run at the speed of light towards the stage, right? So this is to help us to remember that, oh yes, these drawers, there's something that we have to learn about the microscope, right? Microscope. And also there's this scientist called Robert Hooke, right? So this is the anchoring point of your drawer number 1, right? Drawer number 2, plant and animal cells. So new mom cooks meals with veggie curry. So this is your new mom, and she's cooking. Yeah, this is the fire, this is the pot. If you can see, if this is if my drawing is good, okay, this is supposed to mean the fire and the pot, and there's a leaf here to represent the vegetable. And curry, I do not know how to draw it, but you can imagine this is the pot of curry, right? And that is to explain the components of the cells, whether it's the animal cells and plant cells, right? Specialized cells. Okay, this is the funny one. The bloody, so this is sperm, and this drop are the blood to show that this sperm is full of blood. And this is the eggs. He needs the egg. However, he left his hair, the only hair, at home, right? So bloody sperm needs egg, left his hair at home, right? Diffusions. Diffusion is glucose. This boy, his name is Glucose. He is breathing and to exchange gas. O2 is oxygen. So this to represent his breathing to exchange gas. Then drinks water and sleep. Right. So drinks water is to represent water absorption, and sleep is wilting. Right. Next, unicellular organism. Eugene and Angmo. So there are 2 boys or 2 girls. No, Eugene is a boy name. Right. Eugene and Angmo. Or Eugene is an Angmo. Right. Angmo, whoever who doesn't know what is Angmo, is a term that Hokkien people call Westerner. For Malaysian, for sure. For Malaysian who study in who live in Malaysia, for sure you know what is Angmo. For the Malaysian who is not studying, stay in Malaysia, maybe you do not know, but yeah, that's what Angmo is. So to represent Amoeba and Euglena, right? So this is the picture. If you can take your pen and paper and at least write this down, write the story down for each drawer, if you haven't done that yet. And if you can draw this quickly, this drawing is not difficult to draw. Draw it out. I'll tell you why is this important. I'll give you 3 minutes. 3 minutes. You can decide to draw or not. It's really up to you. But you have 3 minutes. Everyone have 3 minutes. 3 minutes later, I'll tell you why it is important. So I hope in front of you, you have the story, which is I sell plant and animal that specialize in moving one object, one organ. And then this part that I requested you to write and draw. This step number 6. So on your paper, you should have these informations: the drawings, the story per drawer, and the story that help you to remember the name of the drawer. Still 2 minutes. You decide. You want to draw, you can. You don't want to draw, just relax. One more minute. Ten more seconds. All right. As promised, let me tell you why do I think that it is important for you to draw. All right. Imagine there are 2 elites. The first elite is trying to, after 2 days, okay, after 2 days, there are 2 elites. The first elite is trying to remember or refresh what she learned 2 days ago about cell. And she has nothing in front of her, or maybe she has this step 2, right? Compared to another elite has this drawing in front of her. Which elite do you think that is going to have an easier method to remember what she learned 2 days ago? Obviously, it's elite number 2, right? Because our brain is designed better to remember stories. Our brain is not designed to remember text, right? If the text is having some emotions, yes, it is also relatively easier for you to remember. Remember the bloody sperm story? Remember your mom cooking the new vegetable? Remember the robot is going to snatch the lens on the stage at speed of light, right? If you have a picture, a drawing with the story, and the story is having some emotion in it, then it's perfect. Then it's much easier for you to remember what you have learned, right? To retrieve the information that you have learned after 2 days. Now you still have a fresh memory on what you have learned, but 2 days later, maybe not. 2 weeks later, maybe even know, right? Because we are human, we are not AI. Our brain is not designed to store information forever, right? So this method of studying, the one that I'm teaching you right now, is to help you to be able to retrieve the information faster. So now imagine, Elise number 3, not only that she has this in front, this picture or this drawing in front of her, she have already drawn this picture like 20 times before she go for the exam. And at that time, she doesn't even need to draw the picture in front of her. The picture is automatically appears in front of her. And that is what I call photographic memory. Right? Okay? So that's why I think it's important. The first step, at least you need to draw. Draw the number 1 time. If you do not draw the number 1 time, where are you going to get the number 20 times? Right? Okay? Now, step number 7, answer quiz. So imagine, now I am assuming that you have your drawing in front of you, and if not, at least you have this in front of you written down, and you have this in front of you in your paper, on your paper. Now, quiz time. How do we do this? Read the question, ask yourself which drawer it belongs to. Then before you read the answer, you formulate your own answer based on the drawing in front of you, based on the story in front of you, and based on this reference that I'm going to put it, I'm going to show it here in front of you. Right. So number 1, read question and ask yourself or answer which drawer is this related to. Number 2, make your own answer based on the reference that you have, your drawing, and also this step number 2 notes that you have in front of you. Number 3, read the answer that is provided and then compare with your answers in your brain. If the answer is similar, excellent. If the answer is not similar, that means there is new information over here, which also excellent. You are learning new things now. And take this new information and put it inside your brain drawer. Okay, so let's do this let's do the number 1 with 1 minute. And let's start now. Read the question. Ask yourself, what drawer is this? And then form your own answer. Then compare the answer that you have in your brain, in your mind, with this answer that you have given. Fifteen seconds. All right. So this is clearly drawer number 1 and under this total magnifications. Okay. So let's do 2 and 3 in 1 minute. Let's start now. Ten more seconds. All right. 4 and 5, 1 minute. Which drawer form? Tell yourself which drawer, form your own answer and compare the answer. Okay, 6 and 7. Start now. Ten seconds. Eight and 9. Start now. The last drawer, draw 5. Eugene and Ang Mo. Okay, last questions. Let's do 30 seconds. Okay. Okay, let me remind you why we are doing this. Why we are doing these 3 steps. Number 1, identify the drawers for this question number 1, number 2 until number 10. And then form your own answer, then compare with the answers that is given here. One of the first sessions that we have attended is to compare the smart student and the not so smart student. And there are 3 skill sets. First skill set is, or 3 differentiating factors. First differentiating factors is the smarter student is having a bigger knowledge base. And the smarter student is able to retrieve information faster than the not so smart student, right? So this is to train you to retrieve your information fast, which is the skill set number 2. Skill set number 3 is to be able to identify new informations and then store it into your knowledge base, right? So when you form your own answer and you compare with the answer that's given, if you identify there is new information that is not in your answer that you formed just now, so there is new information. Pick it up intentionally and put it inside your knowledge base. So it's a positive cycle. Your knowledge base become bigger and bigger. Your speed of retrieving the information become faster and faster, and your skill of identifying new informations is becoming sharper and sharper. And then the knowledge base become bigger again, and because you do it so many times, your speed is become faster, and then because you did it so many times, your brain is become sharper in identifying new information, and that is helping you to become a better version of yourself, which is a smarter version of yourself, right? Now we have completed step 7. Now let's continue on the easy part, the video part, right? So let me just open the video.
All right, today we're diving deep, microscopic deep.
Okay, before I proceed, I just want to make sure everyone is able to hear that lady who was speaking just now in the video. Eric? Yes. What about Elise, Chuan, Jaden, and Jovin, just to make sure? Do you hear? The lady was speaking just now. Let me try again.
Into the incredible
Okay,
world.
great. So everyone is able to hear. Now, how do you do this? You have to intentionally identify which drawer. And you only have 5 drawers. Right? Which are the 5 drawers? Refer to your paper, and that is the 5 drawers that you have. So once this lady is trying to speak on a new subject or a new idea or new information, ask yourself, this one is related to which drawer, right? Let's start now.
All right, today we're diving deep, microscopic deep, into the incredible world of cells. These are the tiny, fundamental units that build, well, everything alive. That includes you and me. And you might be surprised to hear that their story starts a lot closer than you think. In fact, it starts right inside your own house. Seriously, take a look around the room you're in right now. See any dust bunnies lurking in the corner? Well, here's a kind of gross but fascinating fact for you. Most of that dust, it's actually made of dead cells. Yep, your dead skin cells, your families, your pets. So yeah, this invisible world of cells, it is literally all around us, all the time. So here's our game plan for this microscopic journey. First up, we're going to see how we discovered this invisible world in the first place. Then we'll look at life's 2 main blueprints. After that, we'll check out cells that have really specialized jobs. We'll also figure out how they move things in and out. That's the great cellular exchange. And finally, we'll meet some amazing creatures where just one cell is the whole shebang. Okay, let's jump into our first section, seeing the invisible world. Think about it. How on earth did we first discover these things that are completely hidden from our naked eye?
Drawer 1. Huh?
So first things first, let's get a clear definition on the table. What is a cell? Simply put, it's the most basic building block of life. The best way to think about it is like a single Lego brick. It's the smallest possible unit you can have that's still considered alive. Some organisms are just that one single brick, while you're a massive, complex skyscraper built from trillions and trillions of them. And this whole journey of discovery, it all started way back in 1665. A scientist by the name of Robert Hooke was messing around with a pretty primitive microscope, and he decided to look at a super thin slice of cork. What he saw were these tiny, empty, box-like structures. They reminded him of the smaller cells that monks lived in, and boom, the name stuck. That microscope, of course, was the key that unlocked this entire invisible world. And if you've ever used one, you know the basic drill, right? You start low. Put your sample on the stage, use the lowest power lens to find what you're looking for, get it roughly in focus, and then you start cranking up the magnification to see all the incredible detail. But how do they figure out how much they're magnifying something? It's actually super simple math. You take the power of the lens you look through, that's the eyepiece, and multiply it by the power of the lens pointing at the sample, the objective lens. So if your eyepiece is 10x and your objective is 40x, well, 10 times 40 is 400. You're seeing that object at 400 times its actual size. Pretty cool? All right, so now we know how to
Drawer
see
2.
them. The next question
The
is,
2 items that
what
you're
are we
selling.
actually looking at? It turns out for most of the complex life on this planet, things are built using one of two main blueprints. We're talking about animal cells versus plant cells. And you can see the difference right away. Over here, we've got some human cheek cells. See how they're kind of roundish and, well, a bit blobby? Now look at these plant cells from a leaf. Totally different, right? They're much more rigid and rectangular, almost like they're little bricks stacked neatly in a wall. And that visual difference, that's a massive clue about what they're built to do. So what really makes them different? It all comes down to the parts inside. Now, they share a lot of the same basic machinery. Both have a nucleus, that's the brain of the operation. They both have cytoplasm, which is the jelly-like stuff filling it all up. They've got a cell membrane acting like a bouncer at a club, controlling what comes in and out. And they both have mitochondria, the little power plants generating energy. But plant cells have three extra game-changing components. They have a rigid cell wall for structure, a huge central vacuole that's basically a water tank, and, of course, chloroplasts, the little green machines that turn sunlight into food. Okay, time for our third stop, cells with special jobs. Because here's the thing. In a complex organism like a...
Right, drawer 3, remember drawer 3, the specialized cell, the bloody sperm and the egg story, and he left his hair at home. Yes, that's the one.
human, you can't have a one-size-fits-all cell. Instead, they become specialists, changing their shape and structure to be amazing at one particular task. And a perfect example of this, probably one of the most famous specialized cells out there, is the red blood cell. This little guy has one job and one job only, to be an oxygen delivery truck for your entire body. And its whole design is absolutely perfectly engineered for that one single mission. So how does it pull this off? Well, it takes that basic animal cell blueprint and makes some pretty radical changes. For starters, it actually ejects its own nucleus. It just gets rid of the control center to make more room inside for hemoglobin. That's the special protein that oxygen molecules literally stick to. And that weird flattened donut shape, that's not an accident. It gives the cell a massive surface area, which makes it incredibly efficient at both grabbing oxygen in the lungs and letting it go in your tissues. And you see this principle everywhere in the body. It's a classic case of form follows function. A nerve cell is super long and skinny, perfect for sending messages over long distances. A root hair cell on a plant has a huge surface area to soak up as much water and nutrients as possible. And a sperm cell? It's basically a streamlined head with a powerful motor on the back, all designed for one thing, swimming fast. The job always dictates the design. All right, that brings us to our fourth big idea, the great cellular exchange. I mean, cells are living things, right?
Right, so this is the diffusions, right? The glucose breathe to exchange gas and drink water and sleep.
They have to eat, they have to breathe, and they have to, well, take out the trash. So how do they get the good stuff in and the bad stuff out? A huge part of the answer is a process called diffusion. And honestly, it's a beautifully simple idea. It's just nature's tendency to balance things out. Imagine a crowded room. People naturally spread out into the empty spaces, right? Particles do the exact same thing. They move from an area where they're all bunched up, a high concentration, to an area where there's more room, a low concentration. The perfect real-world example, burnt toast. We've all been there. That smell starts out super strong right by the toaster, right? A very high concentration of burnt toast particles. But give it a few minutes, and that smell has spread through the whole house. That's diffusion in action, and it's the very same passive, effortless process that lets things like oxygen seep. And you can actually see the result of this process with plants. When a plant gets enough water, diffusion pulls that water into the cells, filling up those big vacuoles we talked about. This makes the cells firm and turgid, and the plant stands up straight. But if you forget to water it, the water leaves the cells, the vacuoles shrink, the cells go all floppy, and the whole plant wilts. It's a direct, large-scale sign of what's happening at the microscopic level. Okay, for our last stop today, we're going to check out a world where one single cell is the entire organism. We're talking about unicellular life, and these little guys are living proof that you don't need to be big and complicated to be incredibly successful. Let's compare two classic examples. First, you've got the amoeba. It's basically a shape-shifting blob. It doesn't have a fixed form. It just kind of oozes around and swallows its food whole. Then you have the euglena. It has a more defined shape, and it gets around by whipping a long tail, called a flagellum. But the craziest part? It has chloroplasts. It can actually photosynthesize. so it's part animal, part plant, a truly wild little creature. So how does a creature that's just one cell make more of itself? It's a process called binary fission, and it is brutally efficient. The cell just gets bigger, duplicates all its important stuff inside, and then splits right down the middle, creating two perfect identical copies. No fuss, no muss. Just simple, direct reproduction. So there you have it. We've gone from Robert Hooke's first peek at a piece of cork all the way to the specialized little machines running around inside our own bodies. The one constant theme is crystal clear. Cells truly are the building blocks of life. But that brings us to one last mind-bending idea to leave you with. Think about it. A single cell, like an amoeba, can do everything it needs to survive. It can hunt, it can eat, it can move, it can reproduce. It's a completely self-sufficient life form. Now consider the fact that you are made of trillions of cells, all working together in this unbelievably complex, coordinated society. If one cell can do all that, what does it say about the miracle of cooperation that is, well, you?
All right. So we can do a lot more than the amoeba. Now, next step is listen to the podcast. Right. Before we go there, let's have a quick glance again on this step 2. Let's do it for 1 minute. And then let ask yourself if you understand this chapter more and more. Ten more seconds. All right. So I believe nothing changed here. I believe you understood this content or this step 2 much more than 2 hours ago. 2 hours ago when you started reading it, maybe you feel a bit worried. What exactly is this? And I'm not sure what is this. And so those anxious thoughts. But now, when you read it again, nothing changed. I did not change anything here. The only thing changed is 2 hours later. 2 hours later, by following this study method, and your understanding about this chapter become much stronger, right? So we have one more steps for podcast. Let me try to open this file.
Welcome to the deep dive.
Okay, tell me if you are able to hear this guy, this man who is talking just now. Just to make sure everyone can hear the podcast. Let me try again.
Today we're jumping right into, well, the
Can
absolute
you hear them? Eric? It is? Chen?
Yes.
Are you able to hear? Yes. Okay. Yep. All right. So how are we going to do this is I'm going to highlight the parts where this person is talking about, right? And it's going to be, usually it's going to be in sequence. And it's about five minutes. Let's start now.
welcome to the deep dive. Today we're jumping right into, well, the absolute fundamental building blocks of all life.
We're talking about cells. It's really where all of biology begins.
Exactly. We're going to explore everything from their basic architecture to some really bizarre adaptations that keep everything running.
And it's a story that starts about 350 years ago with Robert Hooke looking at cork and seeing these tiny room-like structures. He called them cells, and the name just stuck.
And to see them, we obviously need tools, microscopes. But it's not as simple as just putting something under a lens, is it?
Not at all. The sample has to be incredibly thin for light to pass through. And, a lot of the time, everything is just transparent.
So you have to stain them, add colored dye.
Exactly. That's the only way you can really differentiate between, say, different types of blood cells. It makes the specific part stand out so you can see what they're doing.
Okay, so once we're zoomed in, what's the first thing we notice? I guess the basic blueprints.
Right. And the most obvious difference is between plant and animal cells. Plant cells are very regular, almost boxy. They fit together like bricks.
And animal cells are more irregular, more fluid in their shape.
But they do share a core operating system. Both have a nucleus, which is kind of the control center with all the genetic material.
And a cell membrane, the gatekeeper, controlling what comes in and out.
And then you have the mitochondria.
The powerhouses. Everyone knows that one from school. They handle respiration, transferring energy.
They do, but what's fascinating now is how scientists view them. Poor mitochondrial function is seen as a huge clue, an early sign of aging or disease in a cell. So it's more than a power station, it's a health indicator.
So what about the things only plants have? There are three big ones, right?
Three big ones. First, the cell wall. It's outside the membrane, made of this tough stuff called cellulose. That's what gives plants their strength and support.
And then the big vacuole in the middle.
A very large central vacuole. Yep, it's full of cell sap, and the pressure of that sap keeps the cell firm.
And finally, chloroplasts. That's where the magic happens.
That's where photosynthesis happens. They have chlorophyll inside, which is the pigment that traps energy from the sun. It's an incredible adaptation.
Speaking of adaptation, let's get into specialization. This is where you see how a cell's structure is just perfectly built for its function.
Absolutely. Cells will literally change their shape and their internal parts to get really, really good at one specific job.
So like a nerve cell.
A perfect example, a neuron. It's incredibly long and thin, with all these connections at the ends, basically designed to be a biological wire, transmitting electrical signals around the body.
But I think the most counterintuitive one has to be the red blood cell.
Oh, for sure. It's a fantastic aha moment in biology, to be as efficient as possible.
It just gets rid of its nucleus.
Yeah.
Throws it out completely.
It does. It jettisons the control center, all to maximize the internal space for hemoglobin, which is what carries the oxygen. That flattened disc shape also gives it a massive surface area.
And we see that same principle, that surface area idea, in plants with the root hair cell.
We do. All those tiny hairs just create this huge area to absorb water and nutrients. But what's interesting is what they don't have.
No chloroplasts.
Right. Why would they? They're underground, there's no light, so photosynthesis is impossible. It would be a waste of energy to even have them.
So all these cells need to get things like glucose in and waste like carbon dioxide out. How does that happen?
It happens through a really fundamental process called diffusion.
Which is just particles moving from where there's a lot of them to where there's fewer of them. High concentration to low.
Precisely. The classic analogy is smelling burnt toast. The smell doesn't just stay in the kitchen.
No, it eventually spreads through the whole house.
And that's diffusion. The particles spread out until they're evenly distributed. In plants, water moves into the root hair cells from the soil simply because there's a higher concentration of water in the soil.
And that's why a plant wilts. If the soil dries up, that water concentration difference is gone, the vacuole shrinks.
And the cell goes floppy, exactly.
So let's end with the simplest forms of life. Unicellular organisms.
Just one single cell doing everything, things like bacteria. And a great example is the amoeba. It has no fixed shape at all. It just flows around.
And it eats by literally wrapping itself around its food, engulfing it.
Yep, it forms a little food vacuole. It even has something called a contractile vacuole, which is like a little pump to push out excess water.
Then you have something like euglena, which is, it's a bit more complex, isn't it?
It is. It has chloroplasts, so it can make its own food through photosynthesis. But to do that, it needs light.
So it has an eye spot to detect light.
An eye spot and a long tail, a flagellum, that it uses to swim towards the light. It's this tiny, single-celled, self-propelling solar panel. It's incredible.
So we've gone from these rigid plant cells to the specialized cells in our own bodies, all the way down to the simple physics of diffusion that makes it all work.
It's an amazing journey, from the complex to the seemingly simple.
And here's something to think about. If a single organism, like the euglena, can pack in so many specialized parts, chloroplasts, an eye spot, and a one cell to survive, is it really simple, or is it actually one of the most sophisticated, efficient little machines on the planet?
All right. So let's do the next one. Let's do the MCQ. MCQ. Okay. Create 10 MCQ and ask me 2 questions at a time. Okay. Yeah, before that, imagine that you're in your exam hall now, and what do you have is you already you have your drawings right in front of you in your exam hall. You have you drawn this out. You took 1 minute to draw it out, and now using that as your reference, then you can try to answer the questions, right? So you have two references. One is this one, which we always use as a reference. Number two is the drawing, the drawing that you have right in front of you, or the stories that you have right in front of you if you did not draw, right? So how do we do this? Let's type in the chat. Let's do it in this format. Let's say question 1, drawer 1, answer A, for example. Then we do it like this. If you see in my chat, I put 1/1/A. So 1 is representing question 1, /1 is representing drawer 1, /A representing answer A. So all of you can start to read the question and give me in this format. Give me your answer in this format. So I got the first answer, Eric. Question 1, drawer 1, B. One is B, 2 is. Anyone want to answer? From Eric is B also. Let's wait for Jovin. Okay, Jovin also thinks it's B. Okay, and let's wait for a while. Let's see who else want to attempt to answer. Jaden written B. I assume is question 1. There are 2 questions here. Okay, Jovin also provided 2 answers, same like Eric. Okay. So Xuan and Elise, if you agrees, I'm waiting for Jovin. Yeah. Right, let's see. Okay. Let's do question 3 and question 4. Same way, same format. Which part of an animal cell control the cell and contains genetic materials? Question 3. Question 3, Jaden says B. Which drawer? Drawer is very important because that will help you to reinforce your understanding about the drawer. As I say, our brain needs anchoring point, and that drawer that we created together is the anchoring point for your brain. So question 3, drawer 2, B. Right, question 3, drawer 2, B. Same goes to Eric. Jovin is also, yeah, provided answers. What about Xuan and Elise? Okay. Okay, the question 4, drawer 2, C. What do you think, Ren? Ren is next to me. What about question 3? Do you know what is the answer?
Nucleus.
Okay, you also think it's nucleus. Which drawer?
Two.
Drawer 2. Okay, you remember the new mom cooks meal with green curry? Hi, Ren. Yeah. Hi. Hi, Cheche. Say hi, Cheche, Elise. Right, okay, so for question 4, drawer 2, C. Okay. Now, great. So question 5 and question 6. Right. Which structure is only found in plant cells?
Cell wall.
Cell wall. All right, let's see whether it's cell wall. Okay, remember which drawer is that?
Two.
Drawer 2, right? Okay, question 6, diffusions. Which drawer is that? Remember, you have these 5 drawers.
Four.
Yes. Which answer? I is B. Okay, everyone is still typing. I'm still waiting for the answer. Ren, what's the answer? A, B, C, D?
B.
B? Okay. Okay, Jaden is also B. Okay. Eric is also B for question 5, right? Drawer 2, yes. Yes, Elise, BB. I think your B is representing both. Why so many answers are B? We should ask the AI not to give B as answer. Right, okay, I think everyone agrees that it is B. Let's try. All right, okay, so far so good. Okay, let's next move to question 7. 7, can you read? Okay, which cell carries electrical impulses around the body? Which drawer? Do you remember which drawer?
Specialized cell.
Okay, specialized cell is drawer 3. Okay, yes. What answer?
B for 7 and A for 8.
E and A. Okay, I think Jaden, Eric also got B. Okay. Jovin and Jaden are typing. Let's wait. All right, Eric also got A. Hey, excuse me. Jovin, waiting for Jovin. Okay. Elise A. Yep. Okay, let's try. All right, both correct again. Last set. Question 9. Let's do question 9. What about question 9? Which drawer and which answer?
Draw number 5.
Okay, drawer number 5. Which answer?
B.
B. Okay, what about 10? Which drawer? Your best friend, Eugene.
C.
C. Which drawer? Also 5?
Yeah.
Okay, let's see. Okay, Eric, also we got the same answer. And Jaden also we got the same answer. Jovin also same answer. And hey, Eugene Krabs.
So that's C.
Okay, Jovin and Eric are still typing. Let's wait before we review the answers. Okay, let's do it. Hey, where is it? All right. Congratulations. I think everyone got 10 out of 10. To be frank, this is an easy it's not a hard question. Let's put it this way, right? But it's a good start. You only spend 2 hours, and you're able to understand, I think, quite well this chapter, right? I think good job, great job. Let's continue this way. Let's hope we can meet next week. Next week we will either do the revision of this chapter or we'll learn a new chapter. Let me plan on this, and then I'll let you know. Okay. Thank you very much, everyone, and hope that you have a good weekend.
Bye-bye.
Bye bye.
Bye. Thank you. Bye-bye.
Bye bye.
Bye.
Bye bye.
Should I